All right, so our next presenter is from Aldevron. He is Dr. Todd Howren. He is the Vice President of RNA Client Services. With 25 years of service, Aldevron is a premier contract development and manufacturing organization producing high-quality research and GMP-grade plasmid DNA, mRNA, and protein for the development of vaccines, gene and cell therapies, immunotherapies, molecular diagnostics, and other genomic therapeutics. Todd's going to highlight the new capacity of Aldevron's complete RMNA ecosystem. It offers a streamlined service to develop, produce, and release both mRNA drug substance and product. Welcome, Todd.
Hey, thank you, Perry, and what a pleasure and honor to be joining this forum and having the ability to talk with this group. So first of all, let me apologize. I am coming to you from a hotel room in Boston, so I apologize for the background. But what I'd like to do over the next 20 minutes or so is really just provide an overview of Aldevron, who we are, talk about our capabilities, and then, as Perry alluded to, drill down into and talk about our CDMO services around RNA. So let's jump into it. Aldevron was founded in 1998, so we have over 25 years of experience in the industry in the manufacture of biomolecules. Specifically, if you look in the lower right-hand side of the screen, in plasmid DNA, in mRNA, and in recombinant proteins. To date, we have manufactured over 165,000 batches. And of that 165, nearly 5,000 have been under CGMP or GMP source. Looking at Aldevron as a whole, we have over 325 square feet of facility footprint that is solely focused on manufacturing. One of the questions that we often receive from clients is, what makes Aldevron different from other CDMOs? How does Aldevron really bring value to its clients? And I can answer this question really in two high-level points. The first is we operate in a very client-centric manner. We are hyper-focused on the client's goals. And really, before we enter into a manufacturing process, we thoroughly want to understand the goals of our clients and even down to the molecule that the client is attempting to manufacture so that we can assure that we're manufacturing under optimal conditions in order to release that final product to the specifications and the needs of the client in a fit-for-purpose fashion. And very importantly, in a timeline that meets the client's needs. If you look at Aldevron, we have 3 locations in the United States. Our headquarters are located in Fargo, North Dakota as well. This is where our GMP manufacturing facility is, where we manufacture plasmid DNA, mRNA, as well as protein under GMP. In Madison, Wisconsin is our Protein Center of Excellence. This is where we do all our custom process development work for protein as well as analytical method development. This facility is an ISO 1345 facility. When we manufacture protein under GMP, it is transferred internally to our facility in Fargo. And finally, third, our location in Lincoln, Nebraska. Our Lincoln, Nebraska facility came to us through an acquisition that we made last year of Nature Technology Corporation, or NTC. And this is the home of the nanoplasmid technology, where we do retrofitting of the nanoplasmids for our client-specific projects. If you looking at the campuses or facilities over these 3 cities, what we're showing here is four campuses. The first 2 that you see on the left are in Fargo. Our breakthrough campus is 265,000 square feet. Again, this is our headquarters and our GMP facility. Also in Fargo is our advanced campus, and that is just under 57,000 feet, and that is where we manufacture our research grade DNA plasmids as well as our research grade mRNA. Our R&D team is also located on this facility. Again, then in Madison, that's a 65,000 square foot facility where we manufacture, again, proteins under 1345 and do our custom process development as well as analytical development for our proteins. And then in Lincoln, Nebraska, that's a 65,000 square foot facility where we do, that's our nanoplasmid facility where we do all our retrofitting and design of customer-specific nanoplasmids. So what you're seeing here on the left-hand side of the screen is a picture of our breakthrough campus. Again, this is our headquarters and where we do GMP manufacturing. On the right-hand side, you can see a schematic of the building. There are two wings to the building, a northern wing and a southern wing. The northern wing is, you can see highlighted here, plasmid DNA suites, mRNA suites, as well as protein suites. These are dedicated suites, as well as being dedicated suites to the unit of operation. It's also have dedicated, trained, and qualified operators dedicated to each of those suites. The south wing is where we do much of our GMPS manufacturing or GMP sorts manufacturing. There are administrative offices as well. This is where many of our QC labs and analytical labs are located. Just briefly to provide an operational overview. I won't completely go through this slide, but I do feel it's incumbent upon me to speak briefly to capacity optimization. As many of you may know, last year or about a year and a half ago, Aldevron was acquired by Danaher Corporation. And Danaher has brought to Aldevron some incredible tools that have enabled us to optimize our manufacturing work streams and cut out considerable waste. And what this means for our clients is it's increased the number of slot dates we have available, and thereby increasing the overall turnaround time for the release of a specific clinical program or product. So briefly, just to provide some brief overview of our GMP facility. In our South production train, we have 16 production suites, 6 upstream, 4 downstream, 1 cell bank, 1 fill finish suite, 1 protein suite, and 1 flex suite. In our North production train, there are 10 production suites, 5 upstream, 3 downstream, a cell bank, a fill finish, and 2 dedicated mRNA drug substance suites. Here you can see a picture, just some brief snapshots of our GMP facility. Then looking at GMP source, which is located in our South production train, there are 11 production suites, 4 upstream, 2 flex suites, 3 downstreams, a fill finish, and a buffer prep. And then finally, just looking at our research grade manufacturing facility. So our advanced campus building 1 is where we manufacture our research grade plasmid DNA. We have a capacity within that facility to manufacture over 13,000 lots per year. In our advanced campus building 2, this is where we manufacture our research grade mRNA. And in this facility, we have a capacity to manufacture over 300 lots per year in that facility. Now, turning to the Lincoln, Nebraska facility, where we focus on nanoplasmid, that facility has the capacity to retrofit and design over 400 client-specific nanoplasmid constructs per year. These constructs, after they're designed in Lincoln, they're transferred either to our advanced campus or our breakthrough campus for production under GMP or research grade. In this slide, you can see a few pictures of our advanced campus building 1. This is our research grade manufacturing facility. Turning then to our Madison facility, again, this is our protein center of excellence. where we have the ability to express proteins both in microbial systems as well as in mammalian systems. Within that facility, we have a scale for microbial up to 1,000 liters, and then for mammalian, 3 by 25 liter wave bags. On the right-hand side of the screen, you can see some of the instrumentation and the capabilities that we have within this facility for purification, which are quite broad and vast. Here is a picture of our Madison facility, of the production facility. What I'd like to do in the remaining minutes is just begin to focus in on some of our capabilities around mRNA. As I had mentioned earlier, we have 2 dedicated mRNA drug substance suites. Our second suite just came online at the beginning of this year. And what I'm really pleased to share with this group is that very shortly we will be formally announcing and launching our RNA drug product services, which will include both LNP formulation and fill finish. And I'll talk in just a moment a little bit more about those services that we'll be bringing online. You know, I mentioned experience earlier in your CDMO, and that is ever so true when it comes to RNA manufacturing. Aldevron was involved in RNA, involved with clients with RNA projects long before COVID. We started working first with clients in 2014 on RNA programs. Our first mRNA GMP batch was successfully completed in 2018. To date, we have manufactured, the slide says over 50 mRNA batches in GMP. It's actually closer to 70, over 70 mRNA batches in GMP. And certainly by the end of this year, we anticipate that we will be well over 100 batches, mRNA batches in GMP. These batches to date have been used in clinical programs in phase 1, phase 2, and phase 3. We have several late phase programs in flight with clients right now. In addition to GMP, in 2022 alone, we addressed and manufactured over 250 mRNA batches under research grade in 2022 alone. And most exciting, we are involved in an approved mRNA vaccine program under which we are manufacturing the linear template used in that program. Oh, and here's the poll question. We would greatly appreciate your feedback here. So please take some time to address these questions. So what you're seeing here is an overview, a workflow, if you will, for the manufacturing of mRNA drug substance, all the way from plasmid DNA through cell bank, through fermentation of the plasmid, through linear template, through the IVT reaction and purification, and then finally QC testing and QA review and release. A few points that I would like to just point out on this slide. Number one, within our RNA production workflow, we have the distinct advantage of leveraging our over 20 years experience in manufacturing plasmid DNA for our linear template. And this really provides us with some unique advantages in that we know that when we step into that suite for IVT, that we have a high quality plasmid that is going to yield robust and reproducible results in that IVT reaction. Secondly, we are vertically integrated, if you will, with respect to the IVT enzymes and capping enzymes. Through our protein business unit, we are able to manufacture in-house our own IVT enzymes and our capping enzymes. And what that means is these enzymes are fully qualified, and we understand the capabilities of these enzymes, and we know that how they function under GMP, and we know how they function in a scale-up model. Secondly, it allows us to control the supply chain. So we do not have to rely on other vendors for these enzymes, and we easily control the supply chain and thus the timeline. I should note that we work with other vendors' enzymes, and that's as the client specification. And then finally, just to comment briefly on analytical testing and QC testing. We have a fully qualified mRNA drug substance panel, and that for which we do all the testing in-house. So that really allows us to control the timeline, and we're not beholden to outside vendors for analytical testing and QC data prior to release. Now, briefly, just to quickly touch on some additional capabilities. Speaking of scale for IVT mRNA, we scale all the way from milligram scale in our research grade production, all the way up to 100 grams in the GMP suite. We are fully prepared and ready to scale beyond 100 grams, just pending client need. Briefly to talk on RNA modifications and the bells and whistles needed to be added potentially to an RNA molecule. We have full capabilities in this area, including capping, whether you're utilizing a cap analog or enzymatically capping, whether it's an encoded tail or an enzymatically added tail, and of course, base modifications, nucleotide modifications. Process development is critically important, and when we think about process development, we really you need to approach this from what is the end goal in mind? What does that molecule need to look like? What are the specifications under which that mRNA drug substance needs to be released? And from that, then, it's important to design the appropriate manufacturing scheme. And that's really where we work closely with clients to ensure that once that product is released, it's released as fit for purpose and it's released according to the appropriate specifications. I won't dig into this slide here, but we do have platform methods for mRNA, IVT mRNA, but also we do tech transfer in from other companies. We have a very robust and reliable process for doing that tech transfer method. And then finally, I'd like to just really focus on some of the issues that we have been seeing in the industry over the past couple years as it relates to mRNA programs. And we have consistently seen this challenge from many of our partners. Some of our partners are struggling with the complexity of managing mRNA programs and multiple CDMOs and multiple vendors. We have seen partners that have had several CDMO partners and several vendors. They could have a CDMO partner for the linear template. They could have a CDMO partner for the IVT mRNA drug substance. And they could have a third CDMO partner for the LNP formulation and fill finish. And then layered on top of that, they could have analytical testing vendors that they're also having to work with. So this is a complex logistic nightmare. What we have built and within Aldevron is a complete RNA ecosystem where we can address our clients' needs end to end, from cell bank, plasmid, all the way to drug product, all under one roof, all under one quality system. On the upper half of the screen, I've already talked through the drug substance workflow. The bottom half of the screen is meant as an introduction to our drug product services. And again, experience in this area is critical. And we have, as a part of Danaher, we have immense experience in both lipids and LNP formulation through our sister operating company, Precision Nanosystems. Likewise, we have that similar experience through our other Danaher sister opco, Cytiva or VanRx. And so We are leveraging for fill finish, just to mention, we are leveraging the VanRx SA25 for our fill finish activities. We're already in the works on our first GMP drug product project, and we're already talking with clients with respect to programs that they would like to launch with us. So I will, in the interest of time, what I'd like to say is, with respect to RNA drug product, is that we are initiating and kicking off an mRNA drug product early access program, which will provide considerable benefit to our first few clients with RNA drug product. So I'd like to thank you for your time. If you're interested in learning a little more about Aldevron and our services, or if you'd like to see the complete slide deck, which I skipped through many slides, please reach out to me. I'd like to provide that to you and also get you enrolled in our early access program. Thank you.
Hey, thanks, Todd. Really interesting stuff. So I really like when you described how you overcome, your words, the complex logistical nightmare that our drug sponsors find themselves. Really, really important stuff. Drug sponsors in this space don't have a lot of cycles, oftentimes, at the regulatory aspects of this. Can you help them with pre-IND, NDA, BLA in that area?
Yeah. Hey, Perry, really good question. And, as I stated earlier, it is our goal to be that full service CDMO. We have a regulatory team that is standing by to assist and even help write sections of an IND as needed by our clients. We really like to start very early stage with our clients and really understand their overall strategy. And not only can we help write sections, but we can make sure that we're starting that program even in preclinical. So from a regulatory perspective, we are really greasing the skids, if you will, to enable that IND and then even to enable you on through later phase clinical trials. So that is a service we offer, and we really like to partner with clients in that area.
That's really good. That could be a huge hurdle after you get all of this great work done. That's a huge stoppage. So I have a note that you actually helping with clinical protocols. I don't hear that from CDMOs very much. Is that true?
Not clinical protocols, if you will, within the clinical trial itself, but certainly, protocols or procedures, if you will. If someone comes to us with, let's say, a unique mRNA molecule that they need manufacturing, they have some unique specification that release specification that we require. What we will do, we'll work with our tech ops team as well as our RNA sciences team to design and develop a bespoke, if you will, manufacturing method to specifically be able to release that molecule, release that product according to those specifications. So that is something that we do offer.
Okay, great. Thanks for that clarification. I could have gone down an entirely different route with that one. Thanks, Todd. Great presentation. Thank you.
Thank you very much.